Fantastic. Thanks for having me. My name is Julian Lucas. I am recently just transitioned to the grad program at UC Santa Cruz, but before that, I was the data wrangler for the HPRC. Let me just try and get away from this little bit of light. And so as part of my job as the data wrangler, I made sure that all of the data was on AnVIL, because I think everyone here at UC Santa Cruz has really bought into the idea of that AnVIL is the way to make it so that people can access data in a clean, federated way. And so that was always one of the first things we wanted to do with new data, is try and get it up on AnVIL as fast as possible so folks could use it. And what I'm going to talk about today is just the data, showing you that it's there, but then also how we share some of our workflows that are runnable in Terra and AnVIL as well. Okay, with that, just a little bit about the HPRC project.
Hey, Julian.
Yeah.
Sorry to interrupt. I think we're seeing just a black screen for the slides. Are other folks seeing that too, or is that just me?
Same
oh, did I lose? You know what? I'm going to switch to just a moment. Okay, so this time with feeling. So the HPRC, in terms of what we're trying to do is we're trying to increase the diversity in genomic research. And in particular, we're attacking that at the, in terms of the reference genome type approach. So what are you using when you align data? What are you using when you, actually look at the reference genome? So we have GRCH38, which, obviously was a foundational achievement, but, we all know that it's a mosaic assembly or a mosaic reference where the reference is coming from a number of different individuals, and a lot of it's coming from one individual in particular. And so what we're hoping to do is actually to increase the diversity represented in the reference itself. So that's the call to action for the HPRC. We want to make sure that we have better representation. So as part of that effort, we're going to be sequencing 350 individuals. So, one way to look at the HPRC is as a production grant in a way. We're going to be making hi-fi, ONT, really high quality read sets for 350 diverse individuals. So that data set itself is actually to us very important. And then we're going to be using, sort of automated or semi-automated approaches to assemble those sequences as best we can, and that's something that people in the consortium take very seriously. And then that's another resource. And eventually we'd like to take all that and create, ideally, this is a sort of goal, one, a reference structure that people can utilize. So our initial sampling efforts were within the 1,000 genomes sample cohort, and the HPRC took, they took the 1,000 genomes data that's available and then made sure, especially our initial sampling was just in trios, and then tried to increase try to sample it in a way that optimized the diversity for what we eventually get. So we didn't, just sort of blindly take 100 samples that, were the first 100,000 genomes, or even blindly take it just in terms of, location. But there's actually a dedicated sampling working group trying to figure out what are the best samples to sequence to make sure that we can include as much diversity as possible in the eventual data set and pangenome. So looking at the HPRC, we have maybe 350 samples in a way. It's actually not too many, right? But it's actually a very wide data set. So for each sample, we aim to have PacBio Hi-Fi data, Oxford Nanopore data. We're going to have Illumina data that we a lot of the time just actually grab from the 1,000 Genomes project. Then we have Hi-C data, and for some samples we have other data like BioNano or sometimes StrandSeq, for instance. But we have this core data set, and the idea is that gets released, and even some of the workflows that are related to that get released as part of the HPRC. That data set is also put into phased assemblies. So we take right now we're taking that PacBio Hi-Fi and the Oxford Nanopore data, trio data. We're putting those, we're making high quality sort of reference grade assemblies. And then those get released as well. In our year one production efforts, we released 94 assemblies, which I think when we uploaded them to GenBank, it was like 10% of the high quality current human reference grade, high quality assembly. So it's actually 94 is not a big number, but it is a big number depending on how you're looking at it. In our next round of assemblies that we're working on now, we'll be putting out, we'll be going from 94 to over 200 easily, probably over 300 assemblies. So we released the assemblies and we also released the workflows. I'll talk about a little bit about that. So when we made these assemblies, especially for year one, for instance, we did that work in AnVIL. We took advantage of the data that was in AnVIL. We took advantage of we wrote workflows and made sure that they were available in AnVIL. And then we did the work in AnVIL. So we kind of, ate our vegetables on that one. But it's not painful to us because actually, I think we like Terra and AnVIL here. And so it's actually, when you have 94 assemblies, it's a nice way to do it because of data tables and organization and the ability to not have to copy data. The last step is taking those assemblies and turning them into a pangenome. So we'll talk a little bit about that today, but it's the same story. We're, taking the assemblies, we're making this pangenome structure. You can think of the assemblies as a pangenome, but we can also create what's called a GFA and have one, a file or a set of files that are usable as a pangenome. And then again, the workflows and the data are publicly released. Okay, so how does HPRC use AnVIL? So the first thing, and maybe the most logical or easiest to understand, is that the HPRC hosts its data in AnVIL. So if you sign up for AnVIL, you can go and get the HPRC data and use it. So what we have here is a picture of our public workspace. So this workspace is publicly available to everyone. And we have a number of different tables. Our data is a little funky. It's, I think if you compare it to some other projects which maybe has one CRAM per sample or a CRAM and a set of VCFs, we have a lot of different data for each sample, which, to be honest, can be a little bit confusing. But if you, for instance, wanted every assembly that we created, you can go to the assembly sample table and copy that to your workspace and utilize that right away. And so we think that's kind of hip. Another way that the HPRC uses AnVIL is that we share our workflows in Dockstore. So if you wanted to recreate some of the analysis, for instance, the assemblies that we do, or if you wanted to run our variant calling pipeline, those are available in Dockstore. And due to, with Dockstore's integration with AnVIL, it means that you can just load those directly into Terra AnVIL and run them. And so you can imagine a situation where you create a new workspace, you get the data from HPRC, and maybe you get some data from the 1,000 genome samples as well, for instance, load in workflow from Dockstore, and then now all of your analysis is being done within AnVIL. So today, I'm going to talk about a subset of what's available in AnVIL in terms of compute, and that is, we're going to focus on Terra data tables, workflows, and analyses. So we're doing that just because that's what we happen to do for the year one production efforts. But we have the data, the data available in data tables, which is always really convenient. If you have more than a few samples, it's really nice to be able to run it and then just write your output to the same data table. It makes it very convenient to keep track of things. And then we ran our analysis in WDL workflows. And what that means is that the workflows are portable. So you could run it on Terra, or in AnVIL, or you could run it on your local compute environment. You can run it in the AWS CLI. And that's, important for FAIR principles. But then one of the nice things that you can do in AnVIL is that you can run a workflow based off a data table. And then you can use a Jupyter notebook, for instance, to check the results of some of the workflows. So if you run 100 jobs, you don't want to maybe go through them one by one and check to make sure that they work. You can use a Jupyter notebook to check to download the data from the resultant QC file, for instance, and then you can check it to make sure that each and every one of them works okay. So I put this in because Dockstore is in part a UC Santa Cruz product, so I had to pitch it a little bit. But as I was talking about before, Dockstore, if you're not familiar, is a repository for workflows. And so what that means is if you are, for instance, looking for a workflow and you say like, okay, I'm not very good at WDL, I'm new to it, I want to run BWA mem. This is kind of standard stuff. So you can go to Dockstore and search it. And what that means is you don't have to go to Google and search BWA mem WDL and look through people's GitHub accounts and then try and copy it over to whatever your analysis environment is. In Dockstore, you can just search through Dockstore and see what is listed, hopefully find something, and then you can import that into Terra and AnVIL. And so that's really nice because if you like WDLs are, I think they have maybe a bit of a learning curve, but at first, eventually they become somewhat intuitive, but you don't always want to write a WDL just to do a one-off. So it's really nice to be able to do that. So searchability is really nice. Another thing that's really nice about Dockstore is that its integration with AnVIL and Terra is just like really excellent. And so that means that if you are making changes to a WDL that you wrote, for instance, and then you push that to GitHub, with GitHub integration, it goes to Dockstore, and then it also propagates to Terra. And so that means that you can make a change, and then you can then, without having to really do anything other than push into GitHub, you're able to like see that change in Terra and run it immediately, which is very convenient. Okay. So this is, actually really like this slide. So this slide is showing how the HPRC used AnVIL for year one, kind of very explicitly. So we write our WDLs and our Docker files in GitHub, and then we push them to Dockstore. So the example I'm showing here is our assembly workflow and our QC workflow. So we have our sequencing data, which is a data table which is publicly available that I showed you earlier. And then we run the assembly workflow, which is again publicly available through Dockstore with the WDL workflow runner. And that outputs assembly data. So it's, input from a data table, output to a data table. So you've got all these assemblies. And then while we're, before we go any further, we maybe check the results with a Jupyter notebook. So we launch a Jupyter notebook, read in that data table that we wrote to, and then run through and make sure everything went okay. And then in addition, we have a QC workflow, which is a WDL, which calls a series of tools to make sure that N50 and kind of high level stats like that, phasing, Hamming errors, or switch Hamming errors, QV, high level stuff, make sure that they all work well. And so we run that again on the assembly data table. It's launched in an automated way, all from your web browser, and then it writes the results to the same data table. And again, we can use a Jupyter notebook to kind of read in those results and check them automatically. So the way I think about it, which I think is a bit confusing at first, but in terms of WDL versus Jupyter notebooks, I think things that we think of as being in production or that we are going to run more than once or twice, those become WDLs for us. And then things where we're just kind of checking stuff, or you really have to turn the knobs in order to say, oh, look for this data over here, that's a Jupyter notebook for us. The Jupyter notebooks ideally are as little of the process as possible, because that takes away from reproducibility in a way. But sometimes you can't write too many WDLs. You have to be able to play with the data yourself. Okay, so just to motivate the final example that I'm going to show in the last part of what I'm talking about is what I'm going to show you today is one of the workflows that the HPRC produced, which is a short read mapper to a pangenome. So I'm going to motivate the use of a pangenome really quickly. So pangenomes are created from assemblies. And so here I have 3 haplotypes. And you can see that the haplotypes are share a lot in common, but the bottom haplotype is missing a large chunk of sequence. Okay, and so with a pangenome, what you would do is, in terms of creating a GFA file, for instance, you're going to be collapsing across homologous regions. So regions that are the same, you don't need to represent those 3 times. In this case, you can just say, hey, this is one node. We expect it to be the same for everything. But then you see at the in the middle section, we have 2 nodes. Nodes are just sequences that are created, which show the polymorphism across the first 2. So, but even more than that, actually, even within those top 2, you have actually you have 2 sequence differences. In this case, in SNV across the first 2. So I think if you think at a high level, which is as far as I go on these things, to be honest, you can just imagine the pangenome as a set of nodes and edges where the nodes are sequences, the edges connect them. And I think the thing that's important is to think about, well, each individual haplotype is going to thread through those nodes and edges. And so you can imagine that this is one person here. This is the first assembly that I showed at the beginning here. This is the second assembly, and this is the third assembly. And that becomes important because you can imagine a situation where this third assembly is GRCH38. And so you have a situation where maybe you have a common polymorphism that's totally just missing from GRCH38. So in order to kind of obviate that problem or to increase variant calling quality, John Monlong and others in Benedict Patton's group developed a tool called Giraffe, which in Giraffe is a short read mapper to a pangenome. And then once they have the mapping, they do some tricks in order to make it so that it's available for variant callers. And then they slapped on at the end Google's DeepVariant variant caller. So you've got a really nice variant caller and a pangenome aware short read mapper. So in the example here, you're seeing it's pretty similar to what I just showed, but you're seeing the pangenome and you're seeing reads mapped to the pangenome. And if you're looking at GRCH38 as the green line down here, you can see that once you, like these reads wouldn't map, for instance, very well to the linear reference genome GRCH38, but with the pangenome, you can take that alignment and do what's called a surjection, where you say, okay, now that we have the good alignment, let's project it, or surject it onto the linear reference, and you can see it sort of fixes some of those problems. And then you can take it and now you can call variants on that, which would be very tricky if we were just using GRCH38. Okay. So the thing to know about this workflow, which we're calling Giraffe DeepVariant, is that you can think of it fairly conceptually simply. You're putting in short reads, you're putting in a pangenome, and you're aligning them with Giraffe, and then you're calling them with DeepVariant. The specifics of it, though, are actually a little nasty. So the WDL has 6 tasks just to prepare the data. You've got Giraffe, which is an alignment, so it's going to be a little computationally expensive. So it's 2 tasks, but they're scattered. So they blast out a number of jobs. And then you've got 3 tasks post-processing, maybe like aggregating the data, for instance. And then you've got DeepVariant, which is 7 tasks, but those are also scattered. So you can imagine that this would be like in a local compute environment, kind of tricky. If you were running a shell script or something like that. But the nice thing about WDLs is that John can write a WDL and give it to someone like me, and it should be portable. And then when you add on to that AnVIL, then you can really treat the WDL and even some of the input data as just kind of like one tool. It's kind of like a binary in a way. And so that's very convenient. And that's one of the nice things about WDL and AnVIL is that, there are these things are available, and you don't have to always understand the complexity in order to utilize the tool. Okay, so with that, I'm going to switch gears a little bit, or a lot bit, and I'm going to show you the workspace which we have, which is, all of you can do this in your free time if you like, and which demonstrates Giraffe DeepVariant. Okay. So this is a public workspace that we have available. So I'm just going to search for HPRC, go to public, and you can see that we have a demo workspace available. Okay, so the workspace dashboard has a little bit of information about the workspace itself in order to be self-documenting. So this is just Markdown, which we wrote in so that people can come in if they're not watching a demo, for instance, and learn what they need to do in order to run the analysis. We have a data table, and we actually have, just in case you don't want to run the data, the analysis yourself, we also have a pre-run data table just in case you wanted to look through it. So the first thing I'm going to do is I'm actually going to clone this workspace so that I can have my own personal copy and in order to run analysis. So I click on the three bar icon, and I'm just going to give it a unique name. And then I'm going to assign a billing project. And I'm just going to click clone. Okay. And so when it's cloning, what's happening is the workspace, each workspace is, it's got, the dashboard with the markdown, and then it also has data tables, analyses, and workflows. And so this cloning process is actually taking that data and it's copying over some of it. In like sort of programming language, it's kind of like a shallow copy. So it's pulling over the workflows, for instance, so that you can run the workflow yourself. And it's pulling over the data table, but it's not pulling over the actual data, right? And so that's the kind of fun part for someone like me. So if I go and look at, this is my cloned copy now. If I pull up the sample data table, you'll notice this little icon here. It's saying some files are located outside of the current workspace. So what this file is, so I have this data table and I can use it. I can run this analysis based off this data table, but I'm not paying for storage of this data. And for something like the HPRC data, which can be, it's actually many, many terabytes, that's potentially very important. Okay, so we have this data table, which is pointing to sort of like I drink your milkshake, it's pointing to another workspace over here. What we have here is a sample ID. So this is just a kind of subset sample that John produced so that it's kind of easy to run and illustrative. And it's got input FASTQ1 and input FASTQ2 along with the sample name. So let's get to work. So what we can do is we can run a workflow. This workflow could have been brought in through Dockstore, but we pre-populated it for you in the workspace just so it's easy to follow along. So let's open it up and see what we can do. There are two ways to run workflows in Terra. One is to do it by file paths, where you can just hard code in GS URIs and say like, hey, go get the file from here, go get it from there. The way that I really think is exciting is to do it off a data table. And so you have to choose a data table. So in this case, sample, and then we have to select a sample to run. We only have one right now, so it's not too exciting. We press OK. And then, so now what we've done is we've just said, hey, run analysis on this table on this sample, okay? And the rest is filling in, and you're basically mapping from columns of the data table or, workspace data to the actual workflow. So the first row right here, we're seeing that these are all our inputs, and we're pulling this this input from the workspace data. So this is, maybe in the bucket, or it's pointing to another bucket in this case even, and it's saying, okay, run the workflow with this input here. The kind of maybe easier to illustrate example would be for the sample name input to the workflow, we're running it for this .sample. So if you go back to the actual data itself, this .sample is going to be pointing to HG2 for this particular row. Okay, so all of these inputs you can see are pre-populated for you right now, and so it's particularly easy, and it's you don't have to worry too much about it. So we know that the inputs are probably going to work, and then the outputs are going to write. So this is the outputs from the analysis. And I think the important thing to notice is the output VCF. So this is after all the nasty steps, after all the alignments, after all the variant calling, you're going to get an output VCF from this analysis, and it writes it back to that same data table. So here I'm going to click run analysis. I'm just going to give it a quick description, which is test, and I'm going to launch it. So in Terra, when you launch a workflow, what's happening is that for each task that's run, Terra is going to launch a virtual machine, and it's going to say, okay, well, what data do I need? It's going to copy it over from the workspace bucket to that virtual machine. It's going to run the job, the task that it's in the Docker container that you requested. And then when it's done, it's going to just delocalize. It's going to copy that data out from the virtual machine, which is about to be spun down, and it's going to put it back in the bucket. And so everything starts out and ends up in a bucket, and then that's what the Terra data tables are pointing to. Okay, so you can see here that the status is queued. So I'm going to go ahead and click job history, and I'm going to click on the actual job to see. So you can see that when here the job is running. So I'm going to click on the job manager. And you can see that I'm showing you kind of a pre-baked cake here. you can see that some of these jobs ran in 0 minutes. That's not actually very real. What it is it's taking advantage of something called call caching. So in call caching, Terra is going through and it's checking your workflow, and it's making sure it matches what's been run before, and it's checking the actual inputs. And if those things match a job that's been run before, Terra is actually pretty clever. It's not going to rerun that and charge you the money. It's actually just going to say, hey, let me go look up the output from that value or from that job, and then I'm going to go ahead and just give it to you. Let me just refresh. You can see that the task of caching it's quick, but it's not, it's not instantaneous. And so all of these jobs are showing 0 minutes, but it's actually going through it fairly quickly. And I think one of the things that's really nice is we talked about scattering. You can see, I don't know how scattered this job is. Okay, this is just scattered over one, but there are jobs that I'll run on sometimes that'll have maybe hundreds of different shards. And so this is all shown once here, and you can click on it if you wanted to get details about any of the actual executions. Okay, so I think what we can do now is just sort of pretend that this is done. And then we'll show you what it looks like as an output. And I think, so what we had here is the pre-run sample data table, which has the same input FASTQs, but now we've got the output GAM file, so the graph alignment file, along with the index and importantly, the VCF. So if you wanted, you can click on this and download it to your local drive, to your local computer. You can use GSUtil to download it wherever you like. Or if you want, you can keep this in the Terra AnVIL environment and keep analyzing data there. Natalie and Stephen, do you think I should quickly show the notebook? Spend a few minutes there.
I think that would be helpful. Yeah.
Okay. So let's say you've run this workflow and you're kind of like, okay, cool. Like, what do I do now? Like, what does my data look like? You can do that. Like I said, you can do that wherever you want, but you can see that when I cloned the workspace, these notebooks came over with it. And one of the things we can do is look at the actual data in Terra. So let's click on this notebook here. This notebook has a lot of information about how to interpret the results of the workflow. I want to highlight just one thing, and that is that when you have a data table and you run a Jupyter notebook with Python, you can, and with R, but I'm going to show with Python, you can take the data and read it into a Jupyter notebook, into that environment, and actually play around with it. And so just really quickly, I'm not going to open the notebook, but what we're going to do here is you're going to see that we're, this is typical Python stuff. We're making sure that we're importing all our libraries that are important. And then when we want to actually get the data, the thing that I want to show you is this section right here. So we're calling a library, which we imported above, and we're reading the table. So it's kind of nicely named. We're reading the table to the data frame. And so here we're reading in this sample pre-run table from the workspace that we're currently in, and we're reading it to a pandas data frame. And so if you're familiar with pandas data frames, you know that it's just going to give you a nice, like, indexable or sliceable element of data that you can then work with. And so what we're reading in is not the data itself at this point. We're actually reading in, you can see these are GSURIs. So this is the location of the data, okay, for this particular example. And so what we can do here is, here we're actually downloading one of the VCF files. So you can see we have the VCF, output VCF. And so we're pulling that VCF string of the GSURI. And then in this virtual machine that's running the notebook, we're just copying that data down. And from there, you can do analysis like you would do on your local machine. And I think what's really nice about that is that as you have a workflow, and then maybe you've got a whittle and you don't want to mess with it, you've got results that you want to be able to inspect. You can do that in a Jupyter notebook. If you wanted, you can start plotting with matplotlib or whatever you wanted, or you could even maybe filter the VCF by hand just to play with it. Then you can upload that back up to the data table. And so just as I showed you that you can do table to data frame, you can also do data frame to table. And so I think once you have that, you've got your data in AnVIL, you've got your workflows in AnVIL, and you've got your Jupyter notebooks to help you kind of bridge the analyses or even make data tables. You can really think about how you can do the whole process in one place, which is very convenient. And I think, is there anything else you'd like me to cover?
I think that's perfect. Yeah. Thank you so much, Joey, for this excellent presentation.